Hello everyone, my name is Wang J. I am a computational biologist on human microbiome working at Beijing Institute of Life Science, Chinese Academy of Sciences. My research interest is to develop new approaches to understand the relationship between microbiota and human health. If you are interested, you can get more information through my library page. Here, I'd like to give a brief introduction to our study on maternal and neonatal microbiota. As we know, the human body harbors numerous microbes, which are very important to human health. In recent years, extensive attention has been paid to the microbiota associated with pregnancy and early life because they are very important to reproductive health and our babies. In this study, we are trying to understand how maternal health status during pregnancy shapes the maternal microbiota and also the neonatal microbiota. Next, my colleague, Dr. Wang, will give you more details on our new findings.
Hello everyone, I'm Jinfeng Wang, the first author of this study. We are interested in the relationship between microbiome and maternal and neonatal health. Let's look at it, what we have already known on this topic before this study. It's well known microbiota not only has an important role on infant health in early life, but also has long-term effects. Many factors may affect neonatal microbiota, such as delivery mood, feeding practice, antibiotic intake, or even pet feeding. Then what we haven't known yet? The factors and the microbial variation mentioned above are all exist after birth. Is there anything happened during pregnancy? I think the first question is when did the neonate contact with his microbes for the first time? The second question is whether the unhealthy flora transmits to the offspring during pregnancy. To answer these questions, we performed the following study. We collected more than 1,000 samples and performed 16S rDNA sequencing. Based on the analysis of these sequences, a large number of bacterial signals were detected from neonatal samples collected within seconds of delivery, suggests colonisation has been taken place in the uterine cavity. The samples from different body sites of maternal and neonatal samples were clustered separately, means body site differentiation has been established in the fetus. The components of the neonatal microbiota from 3 body sites are similar, indicating the fetus is under the same uterine environment. The abundance of the component is different, reflecting the enrichment and shaping of the body site-specific microenvironment. Since we proved the bacterial existence in fetus, we want to see if maternal health during pregnancy would impact on the neonatal microbiota. Before that, we looked into the maternal microbiota of GDM. The microbiome of multiple body sites of GDM pregnant women has changed. Next, we investigated the neonatal microbiota of GDM. Similarly, variations of the microbiome of multiple body sites of GDM suffered neonates have been observed. As both maternal and the neonatal microbiota varied with GDM, we want to know, does GDM cause the same trend variation in neonatal and maternal microbiota? The answer is yes. we found the abundance of many bacteria varied with the same trend. To estimate the potential risks of microbial variation on neonatal health, we also performed WGS sequencing on a subset of meconium samples. The results showed that variations in microbiota associated with GDM may give rise to a range of potential risks to newborns. Here are the new findings of our study. Thank you for your attention.